Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled First in Human Trial Trends, How Far We've Come and Where We're Going with PK Data. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Worldwide Clinical Trials who developed the content for this presentation. Worldwide Clinical Trials is a leading global contract research organization that works in partnership with biotechnology and pharmaceutical companies to create customized solutions that advance new medications from discovery to reality. Their full-service clinical development capabilities include bioanalytical laboratory services and phase 1 through 4 clinical trials, as well as post-approval and real-world evidence studies, all powered by their talented and accessible team of clinicians, scientists, and researchers who bring their firsthand expertise to each clinical program. Their team of 3,000 plus professionals spans more than 60 countries and believes that their collaboration in a culture that embraces diversity, equity, inclusion, and belonging. Everyone plays an important role in making a world of difference for patients and their caregivers. Now, it's my pleasure to introduce our speakers for today's webinar. And first, I'd like to introduce you to Dr. Sherilyn Adcock, Chief Scientific Officer, Early Phase Development with Worldwide Clinical Trials. With a background in pharmacy and clinical operations, Dr. Adcock has dedicated her career to early phase development. She has been an integral part of Worldwide's early phase business transformation from a small clinical site operation focused on generic compounds to the highly innovative business it is today. Dr. Adcock has been instrumental in expanding the early phase business to an integrated 200 plus bed, highly flexible, fit for service clinical pharmacology unit, pharmacy compounding services, specialty pharmacy services, and full bioanalysis laboratory, renowned for its medical scientific foundation and its services, staff, and accessibility. Now, it's my pleasure to introduce you to your second speaker, and he's Dr. Jeffrey Stark, Senior Director, Head of Pharmacokinetics with Worldwide Clinical Trials. Dr. Stark received his PhD. He received his BS in chemistry from the University of Texas at Austin, his MS in chemistry from the University of Florida College of Liberal Arts and Sciences, and his PhD in pharmaceutical sciences from the University of Florida College of Pharmacy. Just has a background in theoretical chemistry and molecular structure, drug design and pharmacokinetics, pharmacodynamics modeling, and he has 20 plus years experience in the pharmaceutical industry and has published one book chapter and more than 60 manuscripts and abstracts. 2023 marks Jeff's 20th year with the Worldwide Clinical Trials Organization, so congratulations to him. And now it's my pleasure to pass the mic and the controls over to Jeff. So Jeff, when you're ready, you may begin.
Okay, thank you, Sonia, for that introduction. One thing that I might add that is even after 20 years, I still love what I do. I've had the opportunity to work on some very well-designed drug molecules over the years and some very creative formulations. Many of those products are on the market today. So I appreciate that introduction. And also thank the attendees. I appreciate your time today. I know that the fourth quarter of the year is hectic in our industry. And right now, I think we're all recovering from setting our clocks back this past weekend, so I know that is a challenge for us to get accustomed to the new time. So appreciate you joining the call today. By way of introduction, I wanted to introduce you to some of the studies that we will be discussing today, the first in human clinical pharmacology studies. Single ascending dose is the first study that's conducted. Technically, the first cohort of the single ascending dose study is the first in human. The next study is typically the multiple ascending dose to assess the different multiple dosing regimens. And then we have another group of studies, the drug interaction, food effect, renal and hepatic impairment, mass balance, that's absorption, metabolism, and excretion. Historically, we saw the acronym ADME instead of AME, but in human studies, we're not really assessing the distribution into tissues as we do in animal studies. So AME is a more appropriate acronym for the clinical studies that we do, and also cardiac safety. As you can see, we love acronyms in our industry. We do speak in full sentences as the need arises. Before we get into the details of the talk today, I wanted to get some feedback from the audience regarding your experience in first-in-human trials, and that will help us customize our discussion today so that you get the most out of the talk.
All right, and I will jump in now and launch our first poll question. We have 2 poll questions coming out back to back, so this is our first one here. So this is done in real time, so your participation is strongly encouraged and very much appreciated. And I'll just leave the polls open just for a few seconds since it's a simple question, which is, are you currently working in phase 1 drug development? So you have 2 options below. It's either yes or no, so just click on one that suits your purpose there. Perfect. Now that looks like we have the majority of our audience has jumped in and participated in this first poll question. So thank you for that. And I'm now going to close the polls and let's take a look at the results. Okay, so in our audience, we have 69% that said yes, while 31% said no. So thank you very much, everyone, for that. And now let's jump to poll question number 2 now. And there you go. Have you observed an increase in the complexity of phase 1 studies in recent years? So the options again are just 2, yes or no. So you know what to do. Click on which one is best suited for you. Okay, and that looks like it's the majority of our audience. Wow, it's very impressive, the numbers that are coming up here. All right, with that, I am now going to close the polls and share the results. Okay, so here are the results of that poll question. 89% of our audience said yes, while 11% that said no. So thank you very much, everyone, for participating in those 2 poll questions. Now back to you, Jeff and Sherilyn, but I'm just curious, anything surprising from the results of those 2 poll questions?
Good morning, everyone, and good afternoon to those of you not in the U.S. at this point. This is Sherilyn Adcock. It was very nice to see that information from the polls, seeing that at least 2/3 of you are working in the phase 1 area, and certainly all of us have experienced over the last 10 to 15 years the changes that have occurred or have been occurring, and even since 2020 we're seeing many more changes. Jeff showed that original slide that showed all the studies that really belong in that clinical pharmacology suite of studies that we do in phase 1. Certainly after the SAD study and usually the MAD that follows that, the arrangement, when those studies, other studies may occur in that ClinPharm suite of studies can really vary from compound to compound, and we're seeing more of these studies combined. Jeff's going to talk a little bit more about that, and again, we do appreciate your participation and look forward to any questions. Thank you. Jeff?
Okay, Sherilyn, you just stole my thunder. That was what I was going to say on the next slide. Historically, the studies that I listed on the previous slide were all standalone individual studies. And today we see a combination of these different studies and no set order. So I might describe some of the trends that we're seeing as hybrid studies. We're seeing food effect cohorts and patient cohorts added to the single ascending dose studies. Sometimes we see concurrent studies. Maybe the multiple ascending dose is started prior to the completion of the single ascending dose.
Yeah, in that case, you're absolutely right, Jeff. And I would say over the past probably 5 to 7 years, we see this more and more with a single ascending dose and multiple ascending doses really occurring in kind of an overlapping fashion. Usually cohort 3, cohort 4 of the SAD is when the cohort 1 of the MAD would start. We found from our timing and our project managers really put a lot of detail into the timelines of these studies. And we found that by combining those, say, at cohort 4 of the SAD, cohort 1 of the MAD starting, we can save up to 16 weeks in development in the early phase.
Right, that's a good point as well. We are seeing that by combining or getting more information in these early studies, that we're decreasing the amount of time that it takes to complete this early suite of studies. We're also bioavailability or bioequivalence. That would be when there is a formulation change or new during the development. That would not be a pivotal or formal bridging study, but it would give some information on the PK of a different formulation. Say if we're going from a solution to a solid dosage form. But today, Cheryl and I will talk about how we use PK data to refine the studies for future designs and get more data faster. So here's the list of the topics that we'll cover today. PK analysis. In general, I will be using the term PK analysis to describe the analysis of plasma or blood samples that are acquired in the conduct of these clinical studies. Go over some basic definitions of compartmental or non-compartmental analysis. And then we'll go into a description of the first in human study, the SAD, how that is conducted, the process for data review meetings, and the decisions regarding dose escalation. Then we'll go into the topic primarily for the webinar today is these hybrid studies, the changes that we're seeing in study designs, and particularly the single ascending dose study. We'll touch on some considerations for the MAD design, how we're using the data from the single ascending dose to customize the design for the multiple dose studies. And we'll discuss the future. That sounds a little ominous, but we're basically going to talk about some of the trends that we're seeing in drug development beyond and some of the additional analysis that can be performed. So since this is primarily an applied pharmacokinetics presentation, I wanted to go over a few PK principles and definitions. I promise not to burden you with too much theory. I know that not everyone loves equations as much as I do. Equations are sort of my comfort zone, so I'm used to looking at both diagram and equation when I go through basic PK principles. Compartmental analysis, you'll see different terms, curve fitting or mathematical modeling. That is when we take a diagram, such as those shown on the right, and come up with a mathematical equation that describes the drug going in, distributing, and being eliminated. Sometimes we see the first drug in, drug out diagram that you see is the simplest model that we can come up with, and that is the equation that is shown below. It includes a first order absorption, first order elimination, and that is the equation that comes out. That assumes that the drug distributes through the body very quickly or instantaneously, but in actuality, it takes time for the drug to distribute through the body, and that's where those additional compartments come into play. They may or may not be related to a true body compartment, but they are used to describe the concentration data that we see. I do have that drug in part of the model that I'm describing. That's for the absorption. Absorption and distribution can be non-trivial. One of the most complicated things I've done with modeling is to describe the absorption for some of the new formulations. It can be very challenging. It involves splitting the administered dose into different pieces and assigning different rates of absorption and things like that. So that can be a very complicated part of mathematical modeling. So although we can use compartmental modeling early in design, there's another option that would be non-compartmental analysis. Non-compartmental analysis is common in most of the early phase studies that we do, and you'll see that abbreviation NCA. Basically, we are extracting information from the concentration time profiles without the need to develop a mathematical model at the time. It's the advantages of doing that. It's fast turnaround, and we can extract information from variable data. As the figure on the right, the simulation that we did shows the numbers are all over the place. It would be very complicated to come up with a mathematical model to describe all of these individual subject profiles. So we can use the non-compartmental approach, not coming up with a model, to get data out of that and use in the further development. So in non-compartmental analysis, I'm going to go through some of the basic parameters, and those are also described in the figures. Cmax, maximum concentration; Tmax, time at which that maximum concentration is observed; the area under the curve. That seems trivial, but there's a lot of information buried under that area, and that's often determined using numerical integration. So I think we're all pleased to find out that someone came up with a reason to do numerical integration that we were forced to use in learning calculus class, and here it is. That's the area under the curve. You'll see the terms AUC last, that is the area under the curve to the time of the last sample, quantifiable concentration, and AUC infinity, that would be appropriate for a single dose in which we take that area under the curve and extrapolate to infinity. That would be the total AUC. The elimination rate constant, that's the diagram on the bottom right. It's normally assessed by taking a log concentration versus time, fitting a linear regression through that line, and that represents the elimination rate constant. The half-life is related to that. Other parameters that are calculated are clearance. These are more conceptual, but clearance and volume of distribution. Clearance is a way to describe the rate at which the drug is cleared or removed from the body, and volume of distribution is the apparent distribution into apparent volume into which the drug distributes into the bodies. Clearance and volume of distribution are usually considered the primary PK parameters, but the area under the curve and the other parameters I have listed are often used to in the discussions of some of the first-in-human studies. So the first-in-human study is the SAD cohort, the SAD study. And again, I said the first cohort of the SAD is technically the first-in-human dose. So that is a major milestone in drug development. What started as an idea or a molecular structure drawn on a piece of paper has gone through all the preclinical testing, and it's now ready to be tested in humans. Most of the SAD studies that we conduct are in healthy, normal volunteers. The primary objectives are safety, tolerability, and pharmacokinetics, usually in that order. Of course, pharmacokinetics thinks everything, as pharmacokineticists, we think everything is about us, but that is not the case when we do these first-in-human studies. Safety is paramount. Basic
Yes.
studies. Go ahead.
Yes, yes, and certainly, yeah, from the standpoint, we do appreciate that the PK is important in these studies, but clearly the safety of the individuals is paramount. We have at our facility, we have a clinical safety lab on site. Oftentimes, facilities may use an off-site lab, but usually it's one that will allow the timing, and we are right in the same building, so we are able to do safety labs both for screening purposes as well as throughout the first in man study or any study. And the data is actually downloaded electronically so that our investigators can see it and review it, identify any trends that might occur. And we can use that data in conjunction with the pharmacokinetic data that Jeff and his team create to move forward with the study, as Jeff will describe here shortly. Thank you, Jeff.
All right, yeah, and the basic study design is a double-blind study. So the subjects in the clinic staff are blinded to prevent any bias during analysis or conduct. The drug will be assigned by an unblinded pharmacist who will just provide the drug or placebo to the correct subjects. Typically, in our studies, we see 8 subjects per cohort. That varies, though. We've seen larger and smaller cohorts in single ascending dose studies. 8 seems to be the most common, with 6 on active treatment and 2 on placebo. 5 cohorts is very common as well.
And sometimes we'll see sentinel cohorts within each of these cohorts, basically a subcohort where we will initially dose 1 active subject and 1 placebo, so 1 and 1. And we may wait 24, 48 hours to assess the safety and the tolerability in those individuals. And then we'll dose the remainder of that cohort before starting the dose escalation. So we would dose 1 and 1, wait 48 hours or so, then we would dose the 5 and 1 for the next dose. We've also even split it up further if it was necessary, depending on the number of procedures, the complexity of those procedures, and any concerns that we might have regarding the safety. Jeff?
Yeah, that's a good point, Sherilyn. I think that most of our studies right now do have sentinel dosing. That is a very common trend right now.
Indeed.
That's the safety, yes. So 5 cohorts, I put plus or minus because we may not complete all the planned dose levels in a single ascending dose study, and it may be possible to go beyond the planned dose levels. So we can cohorts have been added.
Indeed, I think probably the record we hold right now is 13 cohorts in the SAD, and we were trying to push it to find the tolerability. So 5 is kind of an average of what we see, certainly.
And after the studies conducted, there is a data review. Sometimes you will see a safety review or safety committee. Different terms are used for that. But the data from the clinical safety and PK are often reviewed prior to subsequent cohorts in the single ascending dose study.
Yeah, and that's really a team approach. We certainly want your from a sponsor side, your medical, your clinical expertise. You're the one that understands the drug compound the best. And then from our team, we would include principal investigators, the project manager, Jeff, or one of his team members that was assigned as the pharmacokineticist or lead kineticist for this particular study. And likely, either myself or other members of the team that are really the boots on the ground at the clinic and your eyes and ears as we proceed with these studies and collect the data.
Right, and the data that are part of those meetings include the safety in tolerability, different clinical observations, available PK data. Sometimes, depending on the turnaround time and the lab and the number of samples that are required, we might see the data for the PK is one cohort behind. But most of the time, I think we're seeing for a given cohort, we're assessing the safety and the PK data together. The non-compartmental parameters that are used in those data review meetings are usually Cmax and AUC. Again, those are the exposure parameters. And the time to maximum concentration, half-life, clearance, and volume and distribution are also discussed. Usually those are presented as summary statistics, mean, standard deviation, and range. But sometimes we will discuss individual subject data without any subject identifiers. The reason that might be important, I've done a simulation and that's shown on the right. If you look at that data set, it looks like the subjects are separated into two groups. That would not be apparent if we were only looking at mean data. So the range and the distribution of subjects in a cohort may be of interest. So sometimes we do add individual subject, but blinded. It's very cautious. We're always sure to be sure that there's nothing in that data set that would unblind the study. Considerations in the data review meeting. Is it possible to proceed to the next planned dose, or do we need to stop the study? Again, Sherilyn has mentioned some of the safety concerns. Area under the curve. Most of the time, I guess it's most of the time, there will be stopping criteria in a protocol, and that's related to the maximum exposure that's determined from preclinical data. So we'll assess that. For later cohorts, we will consider if we're seeing linear PK, meaning double the dose, double the area under the curve. That will help guide the next dose levels. We'll look for anomalies or high PK variability. Simulation that's on the right certainly looks like the numbers are all over the place. What are we going to do with that? Is that high concentration real? Is that a case of extensive or poor metabolizers that may not be known? But we might need to look into that further. That next bullet point, is it necessary to repeat a cohort, either for clinical or PK reasons? Such as if there's an adverse event that was of concern, it may be advisable to repeat a cohort to see if that is indeed what we're seeing across the population at this stage, or if there's an anomaly. Is it real? If we have a high concentration and there are safety concerns with proceeding to a higher dose, we may need to repeat a cohort to get additional data. Sometimes it's possible I mentioned stopping criteria that's based either on the AEs, the PK, meaning the maximum dose level or exposure per the protocol, and sometimes we see PD stopping criteria. Was the target PD effect achieved?
And I think it's important certainly to have the stopping criteria in the protocol. I think it's equally as important to make the stopping criteria reasonable so that you don't paint yourself in a corner and end up having to stop for reasons that are not necessarily appropriate for the study. We're happy to have our SMEs, include our subject matter experts, including Jeff and others, take a look at this and help with that. We see probably 6 to 10 of these types of studies a year. And so we see some of the wording and some of the chemistry and data behind some of the stopping criteria. So we're happy to help in reviewing that to ensure that you don't stop prematurely on your study.
Right, and from a PK perspective, we might need to to consider, is the formulation correct or route of administration appropriate? We have done some very early studies with formulations that did not yield much quantifiable data. The drug just was not being released from the formulation, and so there was some formulation work during the study. Also, the PK blood sampling. I've circled this. You can see the variability. It looks like the time of maximum concentration is spread over a large range of time values. So did we have sufficiently frequent sampling at early time points to capture the Cmax? And did we sample long enough to get a good estimate of the half-life or extrapolate the AUC infinity?
With regard to formulations, we've had studies before where the drug, we didn't even see any quantifiable PK, and it turned out the drug was just degraded in the stomach and did not ever get to the small intestine where drugs are normally absorbed. We took that particular capsule that we had and we enteric coated it. It was a rudimentary enteric coating, but we did enteric coat it, and we found that we were able to get some quantifiable PK. Now that caused the client to stop that study, go back and reformulate. So we were able to find that out early. And that I think was very helpful. Now you can get certain capsules that provide an enteric coated. We can fill those capsules for these first-in-man studies with very exact doses, and we can adjust those doses as needed during the across cohorts as we evaluate the data.
Yeah, and regarding the quantifiable data, we'll also consider the bioanalytical range, the validated range for quantifying or the sample analysis. Do we need to change the lower limit of quantitation? Is our assay sensitive enough? Or do we need to either raise the upper limit of quantitation, or is a sample dilution needed to bring the concentrations into the range that has been validated? So trends in SAD studies. So we're applying preliminary PK data to assess the food effect, content and timing, renal clearance, collecting urine data, plasma protein binding, influence of BMI, sex, age, etc. on the PK. We're seeing a trend for patient cohorts in these first-in-human studies. Those are primarily proof of concept. Does the drug do what it's intended to in a patient? And that's primarily from the assessment of biomarkers or surrogate markers for the effect. Some of those involve CSF collection. And Cheryl, do you have anything to add on that before we get to some examples?
Well, Jeff, we do have one question that I'd like to pose to you from one of our individuals here. Let's see. To calculate NCA parameters for regulatory submission, do you recommend any software other than Win-Non-Lin Phoenix, like R, or an in-house software like using Excel?
I do not think that there is not a regulated software, or the FDA certainly does not have a software that is stated. Win-Non-Lin is certainly the most common, and it is a validated software. I think the problem with some of the other approaches is that there may not be validation for that, and that's been a concern. I don't know that how that is viewed from a regulatory perspective, but when I first started doing this type of work, I had been doing modeling, and then I got into doing non-compartmental analysis. I didn't trust the software, so I was doing all the calculations with a subroutine in Excel. Of course, that's not a validated process, but I was able to show that Win-Non-Lin was correct, and I increased my efficiency by not doing it both ways on the next study. So.
Thank you, Jeff. So
go ahead.
with regard to the recent trends in the SAD plus studies, in addition to blood or plasma PK, we certainly have a number of studies. I would say it's probably not more than about 50% that also have urine collection data associated with them. And certainly cerebral spinal fluid for some of those products, as you mentioned, that are CNS therapeutic areas. We have not had problems with including healthy volunteers in some of these CSF collection studies. You would think that would, it's a rather invasive procedure, but there's very good procedures in place that we use anesthesiologists to help us with those procedures. They make the subjects very comfortable. We've done these actually in young healthy volunteers as well as older individuals, older adults in studies, and even in some of the patient cohorts. And one of the most recent trends we've seen, which Jeff, I forgot to tell you to add it to our little slide here, is biopsies. And that's not unusual for us to see. We've probably done 6 studies this year where we've done some sort of biopsy, either fat pad, a skin biopsy, or such. So that is an area that we're certainly going to watch. And a year from now, if we present this again, we may be talking about that. We do have one other question that came up regarding do we develop assays for PK/PD? We do have a bioanalytical lab in Austin, Texas. It's actually just moved into a new facility that's about 60,000 square feet. We can do both PK and PD. Our platform for small molecule is LCMS-MS, although we can use that for some peptides and others. And we also have a large molecule lab as well. Back to you, Jeff.
Okay, well, let's go through some examples. The first one I've mentioned is that in a food effect cohort during the conduct of the SAD study. Sherilyn has discussed the importance of food effect assessments for a number of years, and we are certainly seeing an increase in the number of studies that we conduct that do have a food effect added to the SAD study. This is actually the trend that prompted the presentation today. So, most SAD studies are conducted under fasting conditions, and although it's not a pivotal food effect, SAD cohorts with a food effect are becoming more common. Just some basic definitions, the FDA and some information from the draft FDA guidance for food effect in INDs and NDAs recommends assessing the food effect early during development to assess the impact on systemic exposure, and is there an increase or decrease in variability. There's also a comment about food effect with different meals, fat, calorie content. I might add that, and I'll comment on this next slide too, is that timing might be important. The basic definitions for a fasted study are fast for 10 years, 10 years, fast for 10 hours, and that's a long fast. And then 4 hours post-dose. Fed, fast for 10 hours, and the meal is started 30 minutes prior to dosing, and then again 4 hours post-dose. The criteria for absence of food effect is stated here, 90% confidence intervals for the fed fasted ratio within 80 to 125, but most cohorts in the SAD study are not powered for this comparison, but we will do a similar analysis during the SAD study just to assess the food effect.
Yeah. And we do use the high-fat meal that's the standard FDA expected high-fat meal for these studies. Even though, as Jeff said, that study just that cohort may not be powered for a full food effect assessment. But that meal, you have to really make sure that your subjects understand what that meal entails, because it's really not what we see now, what people now would normally eat. It's very high in calories and high in fat content, certainly. But we make sure that our subjects understand that it's important to eat that entire meal, and the last bite is taken. We monitor it very closely. First bite 30 minutes before the dose, and the last bite within 5 minutes of dose.
And the some of the trends that we're seeing with the food effect in these early development studies include assessing the meal type. High-fat meal, as Sherilyn just described, a low-fat meal, or maybe a light meal or snack. We've seen that in some studies. Also, meal timing, not just restricted to the time of the meal related to the food effect study, as we just described, but also how can we give a meal? Do we have to do it 30 minutes prior to dosing? What happens if the meal is consumed 1 to 2 hours before or after dosing? We'll assess the changes in the Cmax, Tmax, and AUC. Those are the primary parameters that will be impacted by a meal. And design future studies. Can the drug be administered with food to alleviate GI discomfort? If so, when and what type of meal? So, Sherilyn, you have any comments on that?
Well, as far as the particularly the light meal and snack, we have found, even as we do the SAD study, if we do that cohort with the food effect, and we have like GI upset that's occurring, oftentimes what we'll do is we'll incorporate that, amend the protocol to some degree, incorporate that, provide that light meal in that MAD study where people are likely going to dose from 10 to 14 to maybe more days, so that it's more tolerated, and we can get some good data without causing a lot of distress or tolerability issues within the MAD study.
Right, and also knowing the food effect helps the design of the MAD. If you're doing once daily dosing, it's easy to fast prior to dosing, but if you're doing a twice daily or 3 times daily, that can get complicated. When are you going to administer the meals in those type of studies?
Yeah. Very good point. Very good point.
So knowing that food effect is beneficial. I mentioned some concerns. This is not a major concern, but adding cohorts does extend the time and the duration of the SAD, completing the SAD. And as I mentioned previously, this is not a pivotal comparison, and it may or may not be a crossover in which you're giving the fasted and fed conditions to the same group of subjects. Sometimes we will see this a parallel. We'll add another group of subjects for the food effect cohort. So those are just some considerations, but I'll give one brief example, what we might see. A typical study design, we have a SAD study with five planned cohorts. And in cohort 4, we determine that we're going to do the food effect. So we might have that split up into 2 groups instead of another 8 subjects. So we're going to have the same cohort A would be the same as 4A is going to be the same type of study as in the rest of the SAD study. But cohort 4B, we're going to administer under fed conditions. Typical sampling in our studies, this one shows through 60 hours. We look at the data, we do plot the data, we do PK calculations, and then we do a statistical analysis on those data. And this example, we see that under fasted conditions, concentrations are higher. Under fed, it looks like the concentrations are a bit lower earlier in time, and the peak concentration is observed later. So food delayed the absorption, resulting in lower Cmax and a later Tmax. Does that mean that we need to adjust sample times for future Fed studies? We can look at how we were able to capture those sample times and use this information to better design the pivotal food effect study. From the results in the simulation, there's a small decrease in, there's about a 14% decrease in Cmax. I said that wrong. 16% difference in Cmax and a minimal difference in AUC. So that's very typical of the analysis that we would do on these food effect cohorts. This example is fairly straightforward. To assess renal clearance, we will collect urine data during the study. The typical study design is shown here. Sampling, in this case, we have through 72 hours. That's just based on the PK profile of the drug. Urine samples are often acquired either in 4 or 6 hour intervals during the study. That will be a full urine collection. Urine samples are pooled during each one of those collection intervals. We'll also assess the plasma protein binding. That's important for assessing the renal clearance. If we only had filtration, the renal clearance is dependent on the filtration rate and the unbound fraction. Of course, there's other processes involved: active secretion, reabsorption, and a small contribution to renal clearance based on urine flow. So we will estimate the renal clearance, and I've shown a simulation on the side here, which this is the cumulative recovery of urine as a fraction or a percent of the dose. That's typical of the data that we would present. And then based on these preliminary renal clearance assessments, do we need to do a full renal impairment study that would include normal, mild, moderate, and severe? Sharon, you have any comments on those studies?
Yeah, there are certainly studies that are within that ClinPharm suite. They may occur a little bit later in the drug development process, but they are very important studies to consider, especially for your labeling. And certainly, as drugs are more renally cleared, we have to take into consideration what happens when there is some level of renal impairment. These studies are usually reasonably small, usually 6 to 8 individuals in each of those mild, moderate, and severe categories, with a group of matching healthy volunteers to that are within that same protocol. These protocols may be done at a single site. They may be done, depending on your timeline and your needs, they may be done with 2 or 3 sites, sometimes the moderate and severe impaired patients, and there's very strict criteria for those, are a little bit more challenging to find for these studies.
Okay. We're running a little tight on time, but I did want to go through a couple of special populations and then how we could add patient cohorts in a SAD study. I think the biggest trend that we're seeing right now is cohorts with elderly and obese patients, and that could be because of the likely population that's going to be receiving the drug, or it could be to potential differences in the pharmacokinetics in those populations. There may be a change in the distribution in obese subjects, or there could be age-related decreases in clearance that are important to consider. The criteria for those are very study dependent, but I've listed some general principles there. BMI greater than 32, and the age elderly cohorts has varied in our studies. We've also assessed the difference between males and females with a target 50% male, 50% female in those cohorts. That's a little bit less common. We do look into that, but as far as special populations, obese and elderly seem to be the very, very frequent right now.
Right. And I think it's important to work with any clinical site, particularly our site in San Antonio, Texas. We have quite a large database of individuals. We don't necessarily recommend that you would write in your protocol specifically 50-50 male, female a target, mainly because that brings some challenges as far as each cohort and bringing in equal number of males and females. But certainly our population is about 60-40, 40-60, somewhere in that neighborhood of individuals that have males and females that have participated in studies. So it's pretty common for us to be able to get those. We prefer to see some language that's a little more flexible than exactly 50-50. From a standpoint of older population, the population is getting older, and certainly there are a number of individuals that do not take medications or are considered healthy older adults, and there are maybe limited medications, and we can certainly recruit those individuals up to about 85 years old. From special population standpoint, we have not only done these populations, we have been involved in some rare disease populations. Whereas we ended that last cohort of MAD study, we may have a population of patients, and Jeff is going into that now. Good. I remember the slides, Jeff.
Right. Well, you mentioned that, but I think that as the one of the studies that is in our mind, we were able to add a cohort with subjects with mild dementia, and that was how did the drug perform in patients, and there was assessment of biomarkers and basically a proof of concept. Did the drug work as it was intended, and what did the biomarker data tell us?
Exactly. And we have actually enrolled not only the mild dementia, but some of the rare disease in cooperation with our late phase group that has a rare disease specialty area. And in some cases, we've had to allow for transportation, work with concierge groups to get that last group of patients in with a particular rare disease, but they're certainly extremely compliant individuals and very interested in participating in these types of studies.
One of the other, so that was the study that we were just mentioning. How did the drug perform? Did it do what it was intended in the patients? Another study that we've done recently, and that was published earlier this year, was to address PK concerns, and that was the AME study of a drug in dialysis patients. So we had a full cohort of normal healthy volunteers and a full cohort of patients that were on dialysis. So that was important to assess any differences, because for this particular drug, it's used to treat the discomfort after dialysis, and so that was going to be the intended population. And it was important to determine if there were any changes in the PK profile for that group of subjects. We'll touch on multiple ascending dose. Most of this was already covered in previous slides, but in the multiple dose, of course, you want to dose to steady state. Often we assume 5 to 7 half-lives to achieve practical steady state levels. And then we'll assess the PK profile for the first dose, the last dose, and the trough concentration in between to assess accumulation. The importance of doing those studies, as we mentioned in the previous slide, if there's a food effect, how are we going to conduct the study so that we are assessing the accumulation due to the multiple dosing regimen and not the effect of food? So that's always challenging. And then we'll use that. We can use the data from the SAD study to predict the exposure that we might see during multiple dosing. That can be done from compartmental modeling, doing the mathematical equations as we discussed earlier, doing a non-parametric superposition. That's where you take data, add it to itself, do that until you reach a steady state. And there's also the theoretical accumulation that's based on the average elimination rate. Sherilyn, you have any comments on that? I know we discussed a lot of this on a previous slide, so I'm not sure that we need to spend too much time on it here.
No, I think we're getting close to time, and we probably need to wind up for today.
Okay. So where we're going, the trends that we're seeing are more data faster. We're doing, getting a lot more out of individual studies, and we're also seeing a combination of studies. We've also seen an increase in PK/PD modeling. Although I said that most of the data that we consider in data review meetings are based on non-compartmental modeling, we're seeing a trend for model development, preliminary development of these compartmental models, assessing the covariates that you might need to consider in a pop PK model. Those models are used for different simulations and purposes. I gave a simulation below regarding what happens if you have a non-consistent dosing interval. In practice, TID dosing is not the same as Q8, and how is the profile going to look if we have 3 times a day, not strict 8-hour dosing intervals? So that's possible. We're assessing PK/PD correlation, also developing PD models. I've put some equations on the right-hand side. I will not go into too much detail. The first equation is again the PK model that you would see for first order absorption, one compartment model. The equation below that is the Emax model, typically used for effect for receptor mediated response. I think for those in the industry, we wish that we could go back to such a simple model. Most of the drugs that are being developed have a much more complex effect model, and then taking those PD models and combining them with the PK. We're also seeing concurrent studies. Again, starting the multiple ascending dose prior to completion of the MAD study. That saves time, and those are some of the biggest trends that we're seeing right now. So that is all I have. Sherilyn, do you have anything to add to that?
Well, the only thing I want to add is you all can see now why I like to work with Jeff Stark. Jeff he's the big brain. He's our super smart guy. We've worked together for 20 years, and we could have talked for hours on a lot of this information. We hope we have the opportunity to speak with you again. Please give us your feedback. Tell us what you'd like to hear. Sonia, over to you.
All right. Thank you very much, Sherilyn and Jeff, for that very insightful presentation. I hope everyone enjoyed it. And just want to remind our audience members, you can still continue to send in your questions right now using that questions window. We don't have time right now to answer your questions, but be rest assured that the team at Worldwide Clinical Trials will follow up with you after this presentation. I think we do have time just for one quick question. And this audience member is asking, SAD and MAD are the first studies conducted for a new drug. Do you ever begin other studies prior to finishing the MAD? Let's see if we can get a quick question answer here. Jeff or Sherilyn?
Sherilyn, I know that we started the MAD studies before completing that single ascending dose study.
Yeah.
Are you aware of any other studies that have conducted prior to completing the SAD?
And there's some that may start as the SAD moves forward, probably not concurrently at the very same time the SAD would start, but certainly as cohorts progress. We've looked at drug-drug interaction, as we talked extensively about today, the food effect type study. There are studies that certainly can be added, in certain pharmacodynamic markers that can be added to the SAD study as well as you progress and see what's happening, what the data tells you. You follow the data.
All right, thanks.
Okay, well, thank you very much, Cheryl Lynn and Jeff, for answering that question. We have reached the end of the Q&A portion of the webinar. For those of you who are interested in hearing more from worldwide clinical trials, you can register for their next webinar, Facilitating Investments Decisions for Small to Mid-sized Pharmaceutical and Biotechnology Companies, and that's scheduled for Thursday, November the 30th. And I am going to put the link in the chat box right now so that you'll have it there handy. And there it is. Okay, perfect. And if, as I said before, since we weren't able to answer all your questions, rest assured that the team at Worldwide Clinical Trials will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that you see there on your screen, marketing@worldwide.com. Okay, thank you for that. You will be receiving a follow-up email from X Talks with access to the recorded archive for this event. Additionally, there is a link to view the recording of this event, which you can also share with your colleagues so they may register for the recording here as well. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. So I encourage you to do that. Now, please join us in thanking our speakers again, Sherilyn and Jeff, for that very insightful presentation. Sherilyn?
Thank you, Sonia, and thank you very much, Sonia and X Talks, for organizing this. You did a great job of taking a couple of scientists and trying to get us organized and understanding, how this X Talks seminars work. So we appreciate all the training and support that you gave us. Thank you.
Thank you.
Right.
It's
I second that. Thank you for your guidance and the attendees. Thank you for your time today.
Thank you very much for that, Jeff and Sherilyn. And thank you, attendees, for being with us. That's the end of this webinar. On behalf of the team here at X Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.